Okay, now we're going to look at how DNA actually becomes a protein. And this is called protein synthesis, or the making of a protein. So DNA is like the blueprint, or the code, a secret code. If you guys have ever done one of these used to be a lot of fun back in grade school. That's kind of what DNA is. It's a secret code, or not so secret anymore. It's a code that's going to tell us which amino acid will go in certain spots in order to produce a protein. And remember, proteins are made of long chains of amino acid called polypeptide chains. So they're amino acids that are held together by peptide bonds. And then once they fold into a certain shape, they become a protein. So here's a protein right here. A long chain of amino acids that folds into a certain shape to become a protein. All right, I think we talked about most of this. One thing we need to mention is DNA doesn't go directly to a protein. We don't just go DNA and protein. We have to first form something called RNA. So we're going to have DNA, which gets turned into RNA, and RNA, there's different types of RNA, will produce a protein. And that protein is going to be the observed trait that you see. Okay, so we're going to have DNA. DNA is this right here, double-stranded, looks like a ladder. Uses A, T, G, and C. That is DNA. And then we're going to do something called transcription, which happens in the nucleus. That's the first step of protein synthesis, is transcription. Happens in the nucleus. It's going to make DNA, and we're going to go to form something called messenger RNA, mRNA. This all happens in the nucleus. Now, mRNA is kind of like the secretary that can go into the nucleus, and that DNA is the blueprint. It's locked up. It does not leave the nucleus. It's too important. It's like, I don't know if you're making shoes or something like that, and you've got this special secret recipe blueprint, and you don't want anyone else to know what you put in there. So that will not leave the vault of the nucleus. But mRNA is able to go in, copy down the message on what the DNA says, and bring it out of the nucleus. That's what mRNA is. It's that messenger. And we actually read mRNA in groups of 3 called codons. So this is codon 1, AGC, codon 2 is GAC, and codon 3 is CCC. Now we use those codons to translate RNA, so that message, into a polypeptide. And remember, a polypeptide is going to be chains of amino acids. And those chains of amino acids are stuck together by peptide bonds to form a long polypeptide that then folds into a protein. Okay, so that's what we're going to look at. What is RNA? RNA is single-stranded. It's not double-stranded like DNA is. It's still made of sugar phosphates connected together here, and these bases out here, but it's more like half a ladder because there's no second strand to connect. These phosphate, sugar, and bases are what form the codons that we're going to look at what amino acids will go with. So another difference is there's no thymine. So we have adenine, cytosine, guanine, and instead of thymine, we have something called uracil. Uracil is in RNA, not in DNA, and RNA does not have thymine at all. Okay, so if you're ever looking at a code and you see a U, then you know that's not DNA, that is RNA. There's 3 types of RNA. There's messenger RNA, which we just talked about back here. The one that is able to, where did we talk about it? Right here. The one that's able to go into the nucleus and copy down the code of DNA and bring it out of the nucleus. That's messenger RNA. Then there's something called ribosomal RNA. We've talked about ribosomes before. When we talked about parts of the cell, we know ribosomes are those workers that actually build the protein. Ribosomes are actually made of rRNA. We're going to talk about those more in a minute, but it's where polypeptides are actually synthesized. And then we have something called transfer RNA or tRNA. Transfer RNA is like the key for our code. Transfer RNA is going to be able to connect bases here to the codon on the mRNA, and the other end of tRNA, or the other part of the key, will have a amino acid attached. So it's going to translate from these bases of RNA to a amino acid. Okay, so those are the three parts that you need to know, the types of RNA. Okay, so we need to know how to read the code. Remember I showed you the codon on mRNA? Here's mRNA right here. These three bases form a codon. So each codon is going to code for a amino acid. Amino acids are the building blocks of proteins. So for example, right here we have UGG. If we look at UGG, we see we have tryptophan, TRP. That is our amino acid. And so right here, here's TRP. That will code for it. Okay, so that's how you read one of those charts. Let's look at how, let's look at the different steps of protein synthesis. The first step we kind of talked about already is called transcription. And transcription is where we are making mRNA from DNA. Remember, DNA cannot leave the nucleus. So if DNA cannot leave the nucleus, transcription has to happen in the nucleus. Because we're having mRNA form or copy down that information, that blueprint in the nucleus, and get the code and deliver it to the rRNA, which is in translation. So transcription is just the making of that code, that message, that mRNA. So the mRNA strand is made by an enzyme called RNA polymerase. That should look pretty familiar because we were talking about DNA polymerase. So now we need to make sure that we specify in transcription it's RNA polymerase, which makes sense because RNA polymerase makes RNA and DNA polymerase makes DNA. So keep those separate. And then the other thing that's going to happen is something called a spliceosome is going to remove something called an intron, which I'm going to show you in just a second. The other step of protein synthesis is called translation. Translation is where we're going to make a polypeptide chain from this message that we got in the nucleus. This is going to happen in the cytoplasm or the rough ER. So there's three steps to translation. There's initiation, where mRNA is going to attach to a ribosome or the rRNA. There's elongation, which is where a polypeptide chain is formed. And there's termination, which is where mRNA leaves the rRNA at the stop codon, and you have this polypeptide chain. Okay, so that's the overview of what we're going to look at. Now, let's look at it in detail, starting with transcription. Okay, the first step is transcription. Transcription is where we're going to go from DNA to messenger RNA, but there are a few steps in the middle. The direct transfer or making of mRNA actually starts with something called pre-mRNA because there are pieces called exons and pieces called introns. So there's exons and introns, exons and introns. Exons actually code for expressed traits. Introns, we used to say, to help you think about it, they're in the way. So they need to be cut out. They're in the way. They actually have a lot of very important functions. But we don't have a lot of time in this class to get into that, but they are worth looking into. Introns are really, really neat. For now, we need to get rid of those introns because they are in the way. So first we're going to put a cap on. Remember that's your telomerase caps. Sorry, we have something going on outside. Okay, so we have telomerase caps, and then we're going to use something called a spliceosome that's going to cut out the introns. They're splicing. And you have these intron RNA pieces just floating around in here. And they do have a lot of very important functions. They can even put the exons together in different ways. So that one code can code for many different traits. So that's something to look at later. Okay, and then we end up with mRNA. This is our messenger RNA, which is going to leave the nucleus. So this all happened in the nucleus. mRNA is now able to leave the nucleus and go into the cytoplasm and start step 2, which is translation. So that mRNA piece is the first piece that we need. We also need something called tRNA or transfer RNA in order for translation to happen. tRNA is going to be in the cytoplasm, and they have two parts to them. They have this anticodon part. So this one here has bases on it, GAA, and the other side is going to have an amino acid. So it's the key here. It's floating around with an anticodon and an amino acid. That anticodon is going to find a section of mRNA that it binds to. It matches with. So G goes with C, A goes with U, A goes with U, that one matches. And it is automatically attached to this amino acid. And we need something called rRNA or a ribosome. Ribosome actually has two subunits, which you might hear about in your Connect assignment. For now, we're not going to go into that. But rRNA is very important because this is where our amino acids are connected by that peptide bond. There are certain sites on the rRNA. There's an E, a P, and an A, EPA. A is where amino acid will come in and attach to the code. P is going to be where the peptide bond attaches. So we're going to have to get two transfer RNAs to come, and I'm going to show you this on another slide. And then a polypeptide chain will form between them, a peptide bond will form between them. And then they move over, and this is the exit. So then the tRNA will exit, leaving behind the amino acid. I'll show you that on this next slide. It'll be clearer. Okay, so right here we have mRNA, tRNA with the anticodon and the amino acid, and then we have the rRNA. This is the rRNA or the ribosome. Okay, let's see here. Okay, this is a good slide. So we have anticodon coming in, attaching to the P site, and bringing an amino acid with it. And then we have another tRNA coming in to form at the A site, bringing in another amino acid. So we have two tRNAs bound to this rRNA ribosome with the mRNA message attached. And these two tRNAs brought in two different amino acids. And those amino acids now are going to have a peptide chain formed, or a peptide bond formed between them. And then this tRNA is going to exit, leaving behind the amino acid and forming a peptide chain, a polypeptide chain. Okay, so all three of those pieces are required to form that polypeptide. And then, okay, we talked about termination is where the tRNA is going to leave behind the amino acid, but it can go pick up another amino acid. It still has its anticodon there, and it can bring it back and keep doing this over and over and over. Okay, and this is one mRNA strand. We can make a whole bunch of copies of something very, very, very quickly by having a whole bunch of different rRNAs bind to the same mRNA strand and just crank this out. They're cranking out this polypeptide chain here, or this protein. It's going to fold into a protein. We can make a whole bunch of something all at once. So if you eat a candy bar, we can make a whole lot of protein, right? We can make a whole lot of insulin protein very quickly to take care of that. It's just one example. I'm trying to finish up here so that I don't run out of time. This is an overall summary and pictures for you guys. You have transcription here in the nucleus. We go from DNA to this mRNA, which leaves the nucleus and goes and binds to a ribosome. This is the ribosome with two subunits. Now, once the ribosome is bound to the mRNA, you have the tRNA bringing in amino acids and matching codons with anticodons to form these polypeptide chains. And then we end up with a protein once those polypeptide chains fold up into their proper shape. And then here is a summary again in words that you can look over for your notes. And I did put a whole bunch of videos up, or I guess there's 3 videos from Amoeba Sisters that are really good about replication, transcription, translation. Make sure you watch those. I'm going to have a quiz the next time I put some stuff up on Monday. All right, I miss you guys. Thank you.